So we've covered recording from outside the brain, on the surface of the brain, and within the brain, right? But now let's talk about recording from inside cells. So that is intracellular recording. So on the right, we see the first intracellular recordings from the CNS. Obviously, you can see the action potential. Hopefully now you can recognize it. These are just pictures taken off an oscilloscope. That was how you used to have to do it. And this was done with these sharp microelectrodes, these very fine, approaching the wavelength of light or even less than, like a needle, like a glass needle with saline down the middle that you just stab into cells. Obviously, you can't do that under optical guidance because it's too small. So you just blindly ram it through the brain, and hopefully you go inside a cell, and when you do, you say eureka and you do a little recording. But it's very fiddly, it's very prone to fail. Those tips, it's very easy to make them too small or too big. It's very hard to know what's going wrong when things go wrong because you can't see anything. Enter patch clamp, right? This is what we talked about that Sackman and Nair invented, where on the surface it may look very similar, right? It's still a little glass tube that is heated in the middle and pulled apart, but the difference is the end is a micron or 2 or 3 or 4 across, right? So it's on the same scale, and what this means is you approach the cell squirting out the saline that's in the tube, and then when you get close to the cell, you turn off that pressure that's blowing out the saline, and then the membrane basically sticks to the glass. Now, what you typically then do is you apply a little bit of suction to the back of the glass pipette, which pulls a hole in the membrane, and now you have electrical and chemical access to the inside of the cell. This is called whole cell recording because you have electrical access to the whole cell, but the original way was actually doing things like this, which is where you pulled off after you formed this on-cell mode. You pulled off and got a little patch of membrane. Now, if you do that this way, then the inside of the membrane, the intracellular side of the membrane, is what is facing out. If you go whole cell first and then pull off, then it's the outside of the membrane that's facing out. So this makes inside out and outside out recording. Even the on-cell mode is quite useful. It's like single unit recording, but you know that all of your electrical signals come from that one cell because you're only attached to one cell. And as I said before, Greg Stewart and Dot figured out how to do this under optical control, which is very, very useful. So people refer to this as patch clamp, referring to the patch of membrane. And on the other hand, when most people say I do patching, I patch cells, they mean they do whole cell recording. And then the clamp bit comes from the fact that you either do the recording in current clamp or voltage clamp. Voltage clamp, you control the voltage and record the current, while in current clamp, you control the current and record the voltage. Now, when you record the current in voltage clamp, that's also kind of equivalent to recording the resistance, right? Because you've got a known voltage, the voltage that you're applying to the membrane, you can record some current. So from that, you can calculate the resistance. And it's kind of the same to a certain extent when you're in current clamp. You're recording a known voltage, you know the current is 0, and so you can calculate the resistance. It's a little bit more complicated than that, but let's just ignore that, right? So the beauty of current clamp is it lets you see the real behavior of the cell, right? So Sackman once sort of said, not realizing how kind of silly this was, that real cells work in current clamp. Of course, they don't work in current clamp. They just do what they do, and current clamp is something that's in the electronics, right? But the point is still the same, that cells naturally, their membrane potential fluctuates, and that's what current clamp allows you to see. And so, for instance, this is a train of action potentials that you can record, that I recorded from a cell by injecting current into the cell, depolarizing the cell past threshold, and you get 1, 2, 3, 4, 5, 6, 7, 8 action potentials. you can go further, and instead of just having a single electrode, you can bring in multiple electrodes, right? So what's happening in this picture is somebody is using, here's the electrode on the right here, and they're patching this cell. Here is an electrode on the left, and they're patching that cell. And when they make one of these cells fire an action potential, I'm willing to bet it's that one, because that looks like a pyramidal cell, then in this one, they see a synaptic potential, right? So this is the size of an EPSP caused by one presynaptic cell firing. Not only can we measure the synaptic delay, we see that a single postsynaptic connection or a single connection between two cells is on the order of a millivolt or two. That's actually quite a large one, to be fair. And so using these kind of approaches, you can map out the connectivity of cells in the cortex. And for a period of time, this was kind of the only way of doing it. You might think, well, hang on, can't we do anatomy and see who's connected to who? Well, the problem is that the kind of electron microscope techniques you needed to see whether there was a connection between two cells were so closely zoomed in that yes, you could see a synapse, but you could never trace the axon back far enough to see what cell it came from. Now, nowadays they're making these high throughput screening procedures that allow them to reconstruct areas of the brain, and soon that will be the best way of doing it. Still, probably value for money, this is the best way of seeing if two areas are connected. Maybe that's not even true. There are a few other techniques that might be, but still, this was the way of doing it for a decade. So in voltage clamp, you control the voltage, right? And the reason that this is so advantageous is that trying to understand how an ion channel works in current clamp is really annoying, because let's say you want to know how a voltage-gated sodium channel works. You're in current clamp, so you start depolarizing the cell a little bit to get these ion channels to open. But of course, then the current starts flowing through the ion channels, and that starts depolarizing the cell by itself, changing the voltage you're at, and providing a sort of another source of signal. So essentially, you've got two sources that are changing the membrane potential, the current that you're injecting and the ion channel itself. And so it gets to be very difficult to figure out how the ion channel is behaving in response to a voltage if it's changing its own voltage. So you see your signal is a mix of two things: how much the ion channel is open, but also how the ion channel is affecting the voltage itself. So it's much easier to work in voltage clamp, because in voltage clamp you basically sit and look at this equation here. This is the classical current, conductance, and driving force equation. And by being in voltage clamp, everything in these parentheses is kept constant, because you keep Vm constant, because that's what you set. In voltage clamp, you set the membrane potential. VE, that's the equilibrium potential, that's constant, because that's just to do with the concentration of ions. And so you can record I, and then that tells you G. So you know how many ion channels are open just by measuring I.